The learning objective for this lab is to become familiar with bioreactors, how to run and simulate cell growth with media nutrient solutions, and determine how the monode constant and maximum growth parameters vary based on media used. We also want to understand the batch kinetics of cells and how different parameters may affect cell growth. This will enable us to construct and interpret a bacterial growth curve using bacterial optical density data as well as glucose concentration curves. Bioreactors have many industrial uses, so being able to optimize cell growth with different parameters will make us more adequate engineers in the bioengineering world. As with any type of living organism, E. coli has a life cycle. They initially start in the lag phase in which no cells are being produced because the cells are still adapting to the conditions of the reactor. The next phase, or exponential phase, is when there is the largest amount of cell reproduction. This is due to the consumption of the substrate, M9, which allows for optimal growth and reproduction. The third phase is called the stationary phase, and this halts the increase in the number of cells. The cells will eventually run out of space or nutrients and stop dividing. Lastly, since there is no more nutrients to feed the cells, they enter the final phase, or death phase. The cells begin to die and their population decreases. Equation 1 represents the change in biomass concentration with respect to time. Equation 2 represents the change in substrate concentration with respect to time. Equation 3 solves for mu, or growth rate parameter. As you can see from the equation, it is a function of substrate concentration and the monode constant. Equation 4 is the integration of equation 1. It will be used to plot a relative absorbance versus time plot. From this plot, we can obtain mu max, or maximum growth rate. Equation 5 was derived from taking the inverse of both sides in equation 3. As you can see, it is in y equals mx plus b form and will allow us to find the kinetic parameter Ks from the slope of a 1 over growth rate parameter versus 1 over substrate concentration graph.
When making the media, we measured out 1 liter of deionized water 3 times. "20 grams of M9 salts was then measured 3 times. Each of the 1 liter bottles of deionized water then received the 20 grams of M9 salts. We then ensured adequate mixing. In preparation for sterilization, all glassware, including funnels, Erlenmeyer flasks, and graduated cylinders, were rinsed with DI water and soap. The glassware was then capped with aluminum foil. A strip of autoclave tape was then placed across the foil. This was to ensure that the glassware was not tampered with and kept clean. In addition to the glassware, the bioreactor was also cleaned with DI water and soap. Any opening on the bioreactor was also capped with aluminum foil and had a strip of autoclave tape. This kept contaminants from the atmosphere out of the bioreactor. Enough water was added to the autoclave to ensure that the plate was covered with water. Any glassware used in the experiment was then placed in a basket, which was then placed in the autoclave. We then tightened the lid to the autoclave. We then ensured the autoclave was set to sterilize and not sterilize and dry due to the liquid media. pH probe and dissolved oxygen probe calibration then took place. By the end of day 2, the bioreactor was assembled, the pH and DO probes were calibrated, preparation of inoculation flasks was complete, and all material required for measuring bacterial growth was in place. We now begin E. coli growth in the benchtop shaker. We filled each of the 3 250 milliliter flasks with 100 milliliters of fresh media. We then pipetted 2.5 mils of our inoculated bacteria into each flask. The flasks were then placed on the shaker to begin cell growth. We now began E. coli growth in the bioreactor. Once the media in the reactor is at temperature, we poured 50 milliliters of inoculated bacteria into the reactor. Here is a visual of the reactor running. At this point in the experiment, we see bubbles within the reactor. This is due to aeration of the media. We immediately extracted 1 milliliter from the reactor using a syringe, placed it into a clean cuvette, and measured the absorbance at 600 nanometers. In addition to absorption, pH and DO measurements were taking place. Any glassware that was exposed to E. coli is soaked in a detergent bath for 15 minutes. Any disposable material contaminated with E. coli, such as gloves and pipettes, is disposed of in a designated biowaste container. The objective of the data analysis is to determine the monod constant and the maximum growth rate parameter for both the bioreactor and the benchtop shakers. This was accomplished by using optical density data. When comparing kinetic parameters between the bioreactor and the benchtop shaker, it was found that the maximum growth rate parameter was larger with the bioreactor. The bioreactor had a growth rate of 4.9 inverse minutes, while the benchtop shaker was 1 inverse minute. The maximum growth rate parameters were determined by taking the slope of a relative concentration versus time graph at which the exponential growth phase took place, or the region with the highest slope. The monod constant was determined by multiplying the slope of an inverse growth parameter versus inverse substrate concentration graph by the initial substrate concentration and the maximum growth rate parameter. The bioreactor had a higher maximum growth rate parameter. This was expected as the bioreactor had more available oxygen as it was continuously dissolved. Additionally, the benchtop shakers underwent changes in temperatures and shaking when the flasks were sampled. The monod constant determined for E. coli growth within the bioreactor was 11.9 grams per liter. The monod constant determined for E. coli growth within the benchtop shakers was 2.09 grams per liter. In conclusion, the bioreactor more effectively grew E. coli. This was due to an increased stability in variables and increased oxygen, which is required by the aerobic bacteria.